DNA replication is the process by which a cell makes an exact copy of its DNA, ensuring that genetic information is passed on accurately during cell division. It occurs in the S phase of the cell cycle and involves several key steps and enzymes. Here's a detailed breakdown. 1. Initiation. Origin of replication. Replication begins at specific sites on the DNA called origins of replication. In eukaryotes, multiple origins of replication exist on each chromosome, while prokaryotes typically have a single origin. Helicase activation. An enzyme called helicase unwinds and separates the double-stranded DNA into two single strands, creating a replication fork. This process requires energy in the form of ATP. Single-strand binding proteins, SSBs. These proteins bind to the separated DNA strands to prevent them from reannealing, coming back together, and protect them from degradation. Topoisomerase. This enzyme helps alleviate the tension caused by the unwinding of the DNA by creating temporary nicks and then resealing them, thus preventing supercoiling. 2. Primer synthesis. Primase, DNA polymerases, cannot start synthesizing a new strand on their own. They need a primer. Primase is an enzyme that synthesizes a short RNA primer, about 10 nucleotides, complementary to the single-stranded DNA. This primer provides a free 3-OH group for DNA polymerase to begin adding nucleotides. 3. Elongation. DNA polymerase. Once the primer is in place, DNA polymerase D in prokaryotes or DNA polymerase E and D in eukaryotes begins adding new nucleotides to the growing strand, following base pairing rules, AT-GC. It adds nucleotides in the 5-3 direction. Leading strand. On one strand, the template is oriented 3-5, allowing DNA polymerase to continuously add nucleotides in the same direction as the replication fork is opening. This is the leading strand. Lagging strand. On the opposite strand, the template is oriented 5-3, so the DNA polymerase works in the opposite direction, away from the replication fork. Since it can't continuously synthesize in the 3-5 direction, it creates short fragments called Okazaki fragments. These fragments are later joined together to form a continuous strand. 4. Okazaki fragment processing. DNA polymerase I in prokaryotes, DNA polymerase Y in eukaryotes, RNase H and other exonucleases removes the RNA primers used for initiation, replacing them with DNA. Ligase. The DNA ligase enzyme then seals the gaps between the Okazaki fragments, forming a continuous strand. 5. Termination. Replication fork meeting. In prokaryotes, when the two replication forks meet, replication ends. In eukaryotes, multiple replication forks work along the chromosome, and replication finishes when these forks meet at specific sites. Telomere replication. In eukaryotic cells, the ends of chromosomes are called telomeres, which prevent the loss of important genetic information. The enzyme telomerase extends the telomeres, maintaining chromosome stability. Key enzymes involved in DNA replication.